So what I'm going to talk about today is our status report on Biglycan, also known as TVN-102. This is a candidate therapeutic for all forms of DMD. And it's being developed by Tivorsan Pharmaceuticals. I'm a professor of neuroscience at Brown, and I'm also, by way of disclosure, a founder and a shareholder in Tivorsan. So a little bit about Tivorsan. So our mission is focused on developing and commercializing a safe, effective, and unique biopharmaceutical, TVN-102, or recombinant Biglycan, to treat DMD. And at the very outset, I want to call out how important the partners are in this entire process. So this began, if you read from almost the bottom up, from the NIH. And several institutes there supported this work both in its basic form, with its basic science research, and then the realization that this could be transformed into a drug. Also, critical programs that were in place there. And then the patient advocacy groups are absolutely critical. And PPMD is at the top, and that's not just because I'm here. That's because they have been really the guiding light and the inspiration and enabling this to happen for much of what you've heard at this meeting today. And we are very grateful for that partnership. Let me just overview what I'm going to talk about today. And so this is a slide where you can get our update. I just wanted to make a comment on leadership of Tivorsan. So currently our interim CEO is Michael Myers. Scott Gottlieb was our interim co-CEO through March of 2017, but he stepped down when he was nominated for FDA commissioner, and he was confirmed in May. And I think having Scott on board was critical. He's very interested in orphan diseases in general. I think that will be very good for the whole community. Obviously, he doesn't have any interaction with Tivorsan now, but very important guidance that he provided when he was with us, and in his leadership of the FDA. So TVN-102, where are we now? So we've been focusing, it's been a very good year, on a new manufacturing process. This is a biopharmaceutical, or called a biologic, and manufacturing these is a challenge, and it has to be done correctly. And where we are now is we have a process where we're greater than 95% pure, and that was one of the requests from the FDA in our pre-IND meeting. We've met that. When we got it more pure, it became more active. So there's a threefold upregulation of utrophin. You've heard about utrophin from Michelle, at the membrane in MDX mice as well as in DMD myotubes. Also, there's a tenfold improvement in dosing. So we can get now maximal efficacy at 1 milligram per kg. Before it was 10. And the less of a drug you can give, that's always a better thing. It's also got a very broad dose range. So it's effective in vivo across a 20-fold dose range. That's very important because when we go into the clinic, we want to have as wide a target as we possibly can. We're currently scaled to 200-liter bioreactor. Just to give you a perspective on that, the output from a single 200-liter bioreactor can support an entire phase 1 trial. Okay, so that's making it at commercial scale for testing. And we've improved our yield, and the commercial feasibility is established. And in safety pharmacology, which is really our last step before IND, we've completed our dose range finding studies in monkeys and in rodents. And we're queued up to do our definitive safety pharmacology later this year, with the goal then for the IND filing in the first half of 2018. So Biglycan. So Biglycan would be effective in 100% of DMD boys. It's not limited by mutation. It addresses the primary pathological defect in DMD, which is the destabilization of the muscle cell membrane. You want to get this disease process as early as you can, okay, to prevent all the knock-on damage. It's unique, Biglycan, in regulating not only utrophin, but also nNOS. And I'll talk a little bit about that more in the next slide. And so it directs the assembly of a functional utrophin complex. What Biglycan really does is recruit utrophin to the membrane. And that's important because in biology, as in real estate, it's all about location. And if your protein is not in the right place in the cell, it can't function. And so utrophin needs to be at the membrane. And that's what Biglycan's normal function is. And that's what we have leveraged in terms of transforming it into a drug. We know by systemic dosing it reaches heart as well as skeletal muscle. And it's expected to be synergistic with existing and emerging therapies. It would work on top of or alongside truncated dystrophin produced by exon skipping or gene therapy, and it would complement drugs targeting secondary pathologies such as the anti-inflammatories, anti-fibrotics, and the muscle size enhancers. Okay, the rationale for Biglycan therapy really comes from looking at normal development. And so in normal development, there is utrophin all around the muscle fiber. And early on, there's no dystrophin at all. And then, as we pointed out earlier, what happens is dystrophin comes along, takes the place, and the outcome is a healthy muscle fiber. In a dystrophic muscle fiber, there still is utrophin, but as the myofiber matures, there's not a sufficient quantity at the membrane. It's not assembled properly in such a way that the muscle cell dies. We discovered Biglycan as a molecule whose normal job during development is to localize the utrophin. Okay? And when you took Biglycan away in a mouse, the utrophin levels went down in those muscles, in the young muscles. We said, well, if this is the way the body normally does it, can we use this in a disease situation, particularly, of course, DMD? And so that's the idea of delivering Biglycan now to restore utrophin and nNOS to the membrane. So a little bit about the context in which Biglycan works. So this is a very high-power schematic of the muscle membrane. I've highlighted utrophin on the inside, and then Biglycan is the red horseshoe. And critically, Biglycan binds to two of the sarcoglycans, the alpha and gamma, and those are only expressed in skeletal muscle and heart. So there is, if you will, an intrinsic targeting, okay, also related matrix molecules such as collagen 6. And that's important because we think that's why a single dose can last a long time, because it stably associates with the extracellular matrix. And utrophin, as you heard, will stabilize the membrane, reduce cell death and resistance to damage. But Biglycan has another facet to its mechanism of action, and that is by recruiting and regulating a complex of proteins normally associated with dystrobrevin, syntrophin, and nNOS. And that functionally is important for reducing fatigue, improving contractility, and reducing damage from reactive oxygen species. Intracellular nNOS is a loose cannon, okay, where it needs to be is at the membrane to function and carry out these roles, and this work has been published. So a little bit of data on our early efforts, and this is the T2 Biglycan, which was one of our development formulations. We treated MDX mice for 3 months and at different frequencies of dosing. This was work all done outside the laboratory, outside Tivorsan. All the dosing was done in Sacramento. And then the mice were shipped to the Wellstone Physiological Assessment Corps, directed by Beth Barton, who did all these studies blind to experimental conditions. And I just want to call that out for a moment, because this Wellstone Corps is one node and facet in what you have done and Pat has driven with the MD Care Act. There are Wellstone centers that serve the entire community, and it gives us the ability to have reproducible, accessible expertise, and all of us as we do our development work. And that really comes down to the MD Care Act, and it does much else. But it's really important to think about what your work has done in terms of really supporting many of the therapies, probably most of what we've seen in this meeting. So when we do that, we restore muscle function to 65% of wild type levels. And the curves you're looking at, basically, the key thing is to look at that dotted line across the top. That's the wild type level. And if you look at the blue curve in the top, you can see that dosing there once a week, we get a very significant improvement over the placebo-treated mice. And the same data from the last eccentric contraction is plotted in the lower right. So this is important because this is the muscle damage. So there's less damage to the muscle on use. And as we all know, that's the problem. Muscle gets used, muscle gets damaged, and it dies. This is preventing that damage, and it's dosing for 3 months. And we were able to then get a first read on how well tolerated TVN102 is in MDX mice. And all of these curves look the same, and that's good. because these are mice that have been dosed at the efficacious doses for 3 months, and the organ weights are the same. None of the animals died. Liver, kidney function, also very good. And then we did see an encouraging signal in the heart. So you'll notice that there is a reduction in the heart of this, in the size, weight of the heart in the animals dosed with Biglycan. We know that Biglycan gets to the heart. We've measured that independently. The fact that the heart's getting smaller is a good thing because, as you know, the heart enlarges pathologically. And cardiomyopathy is characterized by an enlarged heart. So a heart that's getting smaller is going in the desired direction. And in the future, we'll be looking at that more directly. Okay, so with that as motivation to go ahead and really bring this into patients, what we had to do was to make the molecule to the specifications that are required for delivering it to humans. Okay, we started with prototype. That's all the data I've showed you up till now. And we learned that it was heterogeneous and it didn't have the best dosing properties, and we needed to purify it and to bring it to heel, if you will. So all those arrows are very much like what you just saw with what Stu just showed you in terms of what that path looks like. It's not a straight line, but now we've learned how to make this molecule, and having done that, we have an optimized clinical candidate. And that work, which was not only important just for the regulatory checkbox, but turned out to be very important in terms of how well the molecule works. So the first thing we found is that we improved our dosing tenfold. So on the left, with our earlier preparations, we didn't see maximal activity till about 10 milligrams per kilogram. That's the red circle. But on the right, now we're using the optimized formulation and purified material. We see maximal activity at 1 mg per kg. So there's tenfold less that you need to get equivalent bioactivity. Again, an important step. Also, it worked better. So one example of that is looking in cultured patient myotubes, okay? And again, from a biopsy. So there are many ways these biopsies can be used. And so we were able to show, looking on the left, that treating these cultured DMD myotubes with Biglycan, we saw a large increase in the amount of utrophin associated with the membrane. It's a TVN-102 treated. And on the right is our standard curve. And from that standard curve, we were able to determine that there was over a 3-fold increase in utrophin in these cultured cells. So with that, we went back to the mice, and we wanted to test now the efficacy of the new formulation, TVN-102. First, we wanted to ask, what about delivering it to symptomatic mice? Okay, when they are in the really critical, the crisis period in MDX mice, which is about 4 to 8 weeks. And so what we did was we delivered Biglycan in 2 doses, as shown there. And then first we looked at the levels of utrophin. And so this is now looking at, from this study, where we delivered either vehicle on the left, placebo, or 1 mg per kg TVN-102. And by eye, I hope you can see that there is a very significant increase in the levels of utrophin that we see in these animals that have been dosed for 2 weeks with TVN-102. And we quantified that. We asked how much utrophin is at the membrane. And looking in now 1 and 10 milligrams per kilogram, and we've seen this up to 20. Now we're seeing, again, over a 3-fold increase in utrophin at the membrane with this 2 weeks. So we're getting very good efficacy in our primary target for utrophin. And also we wanted to look back again at sparing the muscle cells. Of course, what you want to do is prevent the muscle cells from dying in the first place. Okay. So if you deliver the Biglycan before the muscle cells die, at about 3 weeks, 18 days of age, you can then, by counting central nuclei, you can say how much regeneration has occurred. And here you're looking at a dose response curve. So 0 on the left, the empty bar is again the vehicle. And then we see efficacy, really, we begin to see signs of efficacy at 0.25 mg per kg, and we see it out 1, 10, and 20 mg per kilogram. So now we have a very wide dose response which we can work with. And this, again, for the clinic, very important, because one of the most important things to do in the clinic is hit the dose right. And you cannot predict completely from a mouse model what's going to happen in the human. But it's encouraging that we would expect or hope that we would see a similarly broad dose response in the patients. Okay, so then finishing up TVN-102 progress and plans. So what I've told you is we have a highly purified preparation of TVN-102, new manufacturing process, it's scalable, it's suitable for use in producing clinical material. Dosing's improved 10x, utrophin over a 3-fold increase, and it's active across a 20-fold range. With all that data and then the critical safety pharmacology that's coming up later this year, our goal then is to file an IND and enter the clinic in the first half of 2018. And so finally, I acknowledge you as funders and partners, and I thank you again. You are why we do this work, and we will continue on. So thank you very much.